that signed up. As Carol mentioned, my name's Byron Cheatham, and I'm Vice President at CytoViva, responsible for market deployment of CytoViva's microscopy and hyperspectral imaging technologies around the world. And today we're going to focus on how this technology is used for drug delivery, nano-drug delivery related research applications and initiatives. And I'll start by saying that what sets this technology apart from all other technologies related to any sort of nanoscale and or macro level spectral analysis capabilities around drug delivery and drug configuration is imaging. This is an imaging-based system, and it does something that no other tool will allow you to do. It provides spectral data in very high spectral resolution, in very high spatial resolution context. And so let's go over the webinar real quick. We're going to just give you guys just a brief overview of who CytoViva is as a company and the types of research institutes that utilize our technology today. We'll talk some about the technology and how it works. And then we'll dive into not only nano-drug delivery applications, but we're also going to look at how you can do characterization of drugs and excipients in even more micro or macro scale in addition to nanoscale. So as you have questions, please enter them in the chat box today. And when we're finished, we'll go through all of the questions, the Q&A, and answer these to the best of our ability. And if there's some that we can't answer, we'll take that on an offline basis for you to get your answer. We want you to know that CytoViva is born out of academic research here in the U.S. We're affiliated with Auburn University, and we're actually in this building in the Auburn Research Park. And we were commercialized in 2004 based off of imaging technology at Auburn University, which is a public research institute in the southern part of the U.S. here in the United States. Today, this technology is utilized in almost 400 research labs worldwide. And while CytoViva is a smaller company relative to instrument companies, we have a very robust partnership around the world that helps to distribute these technologies in Asia and in Europe and other markets around the world. So we have the ability to deploy these systems and have in every market in Asia, every market in Europe, and of course, North and South America as well. The deployment of those systems has resulted in a lot of publications, and this instrument is also being utilized not just in development and research applications, but also in pharma for more quality control related applications. But this is just a partial listing, a partial bibliography, more as it relates to nanomedicine and nano-drug delivery related applications. These are just some example publications that are out there in the literature that you would see with a simple Google Scholar of CytoViva or CytoViva drug delivery related keywords that you might search on. To give you a little bit more specific insight on some of the types of research labs that utilize the technology, and more specifically what they do with it. The list here on the left side are more national research labs in the U.S., such as the FDA or USDA, U.S. Department of Agriculture, or NIH, the National Cancer Institute. And you see there are a lot of themes related to nanotechnology, nano-drug delivery, and those sorts of things, nanotoxicology. And that's because this technique is really well suited for the ability to quickly and easily observe an image at the nanoscale and spectrally characterize at the nanoscale a wide range of materials. You can see these are more academic institutes here in the center, and then on the right are more private industry related institutes, most of them which are pharma related. And again, you see a general theme of nano involved in many of these. But there are a lot of applications that are non-nano in nature where this is utilized as well. We're going to show at least one example of that. Just at a high level, I want to share with you what the technology allows you to do, okay? The first thing that it allows you to do is to optically observe nanoscale samples. And the brilliant part of this is because of these enhanced darkfield optics, which we'll talk about on the microscope itself, you can observe nanoscale samples, nanomaterials or other inorganic or organic related materials interacting with biological samples, cells and tissue, for example, or with other material matrices, okay? No sample prep is required at all because of the nature of the optics of the microscopy that we'll use. No fluorescent labeling is required, like you experience with confocal or epifluorescence, which only allows you to see a fluorescent label. It doesn't allow you to see the endogenous material itself. And with electron microscopy, there's all sorts of sample preparation that's typically required for that. Now, electron microscopy is the ultimate tool for resolution of nanoscale materials, certainly. And this doesn't replace that. And we don't want to represent that it does. But at the same time, it's super complementary and can give you information on your benchtop every day in a very interesting way. It also allows you to, as we mentioned, observe these nanoscale samples interacting with labeled or unlabeled biological or material matrices. So if you look in this just image here, we have macrophage cells, structures, and these macrophage cell structures have internalized low-density lipoproteins that can be utilized as a drug delivery vector and some other applications, especially in a cardiovascular-related environment. But not only can you see the LDL internalized inside these macrophages in a label-free manner, you can also spectrally identify, spectrally map these nanoscale samples based on the unique optical spectrum. And we're going to talk about what optical spectrum means in just a second. But one of the things that I want to do real quickly is actually demonstrate for you what that sample looks like with a live dark-field hyperspectral image. And so I just want to show this to you to give you a sense of what the system is and how it works. We have two dark-field hyperspectral images here on the screen that you can see right now. The one that's on my left here are the macrophage samples that have internalized the low-density lipoproteins, okay? The image that you see here on the right are negative control macrophages that were not exposed to the LDL. And so just from an optical perspective, you can see that these nanoscale LDL materials have been internalized by these macrophages. You can see them there, and it's clear and obvious the differences just from a qualitative optical perspective, what you're able to see. But because this is a hyperspectral image, a dark field hyperspectral image, what that means is that every pixel, and this is a zoom image of this red square that we have here, of this larger cell that we have, you can see that the crosshair here is on a pixel that has one of the LDLs. You can see that that's here in the big image that we have. This is the optical spectrum that we're looking at there. You can see how the spectrum changes, right? And if I go in and collect spectrum and I come over here to an area off of it, you can see a dramatic difference. This is an area of the cell where there is an LDL, and then we're going to come back here, and then we're back on the LDL again. Every single pixel of this image contains the optical spectral response of that pixel's spatial area. And with the 60x objective that we're using here, pixel sizes here are about 213 nanometers as we blow those pixels up, and you can see those there. And so one of the things that we're able to do as a result of the ability to get on that, right, and capture that hyperspectral images, we can use analysis software in the hyperspectral image analysis capability to do things like map all of the materials that are unique, in this case, to the low-density lipoproteins that we see here. So you see here now, we turned on the channel to map everything red that's a match to the unique optical spectrum of the low-density lipoproteins. And there's a way to build a spectral library to conduct this mapping in such a way so that you can conduct this mapping with 0 false positives. Okay, there's a process for that. We're not going to go into how the image analysis software works this morning, but because of that, I can not only qualitatively observe the presence of the LDL in the cells, I can quantitatively map them based on their unique optical spectrum with a super high degree of confidence. And I wanted to just share this data with you before we get into how the technology works, because it's really good to just sort of see up front the potential of what's possible with the instrument and what you can do with it. And so we wanted to do that this morning. And with that, we'll transition back into the technology itself and provide some overview. This is what the actual instrument typically looks like with a more typical configuration. And you see a basic research grade optical microscope as at the core of this technology. And this research grade optical microscope is a fairly standard microscope with some very unique optics on it, okay, that allow us to be able to see at the nanoscale in a label-free way and allow us to be able to capture the optical hyperspectral data that we showed there. And so you can see that one of the most interesting aspects, and we're going to spend some time on this, are the dark-field illumination optics, the condenser optics that are on this microscope that allow us to get a very high signal-to-noise ratio, as you observed, of the LDL and the macrophage cells. And we're going to talk about how that works. This microscope system can work in transmitted bright field, which is very typical bright field that's used in pathology and related applications. It can work in transmitted enhanced dark field microscopy, which is the optical mode that we were in the sample that I just showed you with CytoViva's patented dark field optics. We can work in reflected mode. If you're working with very large sample areas, or if you're working on an opaque substrate, silicon or similar type substrates, you can work in reflected mode where light comes from the top through the objective down onto the sample. And you can operate in reflected bright field or dark field as well. If there's a need to be able to observe in fluorescence, we can do that as well. And we actually have a fluorescence module that we can include with this system that's super unique, that it allows us to be able to observe fluorescent and non-fluorescent sample elements simultaneously in the same image in real time. And that's super important, especially if you're doing nanoscale studies. Let's say, for example, that you have fluorescently labeled elements of your cell structure, and you want to know how an unlabeled nanoparticle is interacting with those. With CytoViva's dual mode fluorescent module, as we refer to it, you're able to do those things. I'm not going to spend a lot of time on that today, but that's an option as well. And as it relates to the hyperspectral imaging, you can operate in the visible near infrared range, or you can operate in the shortwave infrared range with this system as well. And so with a dual port camera mount here in this configuration, we have a visible near infrared. This is a SCMOS camera integrated into a spectrograph that comprises the spectrophotometer. On the back of this dual port, we actually have an optical camera so that you can capture optical images or videos if needed as well. But this could be configured with a Wiener hyperspectral imager and a Swer hyperspectral imager if needed as well. We capture these hyperspectral images using a push broom or line scan related technique. We'll go into that in just a second, but we wanted to share with you what the system looks like that's able to capture these images, because it's a pretty basic setup from an optical microscopy perspective, but it allows you to do some super special things, okay? And let's kind of go into what allows us to equip this microscope with the proper optics and spectroscopy to get these really awesome images and spectral data at the nanoscale. Well, let's start with the patented enhanced dark field optics. Dark field microscopy has been around for over a century, right? And it produces some distinct advantages. But if you were to purchase off-the-shelf dark field optics from Olympus, Zeiss, Nikon, they're all the same in the configuration and the way they work. They have some very inefficient processes that don't allow for the type of signal to noise ratio based image you get with CytoViva's patented optics. And I wanted the light comes through the base of the microscope and goes through a series of plastic lenses that don't do a very good job of managing that light. Light then goes through a field diaphragm and goes through about 9 centimeters of air before it goes to the dark field condenser. And in order to be able to focus this condenser onto the sample, you have to move it up and down in the z-axis and in the xy as well, or to focus the light on the condenser. But then to focus it on the sample, you then have to move it again. And that whole process is really ineffective for optimizing dark field, okay? And that's the setup. CytoViva and the developers of the CytoViva Darkfield Optics were working with standard dark field and understood its ineffectiveness and its inefficiencies, and they created an improvement in the way that light is managed to a condenser to dramatically improve the signal to noise ratio, the scatter of the material of the sample versus the background, which is dark. And if you have lots of noise in the sample, that noise is coming from the source light. It's light. And so by managing the light in a super precise manner to the dark field condenser, you're able to get lots of photons in a very oblique angle, okay, in a circular angle, such that you have lots of photons here, and you have very few stray photons above and below that. And so as you move this highly oblique angle light, very shallow cone of light, onto a very shallow or thin focal plane of the sample, you're able to get scatter from the sample into the objective, but without lots of noise or interference. And so we see a dramatic increase in signal to noise ratio over what you get with standard dark field optics, about a 10x increase in signal to noise ratio. And we'll just show you what that looks like here. This is a paper that was published by a group in Korea a number of years ago, where they actually did a comparative test of conventional standard dark field. In this case, it was, I think, Olympus Darkfield Optics, but it could have been any of the major microscope companies. They all do it the same. And you see a dark field image of, in this case, of 160 nanometer iron oxide nanoparticles. And here they also, the same exact particles, utilizing the same exact optics, except they replaced the dark field condenser with the CytoViva enhanced dark field optics. And it's intuitively obvious that you get just a dramatic increase in signal to noise ratio between these two, right? Lots of noise in the background, a weak signal. Hardly any noise in the background, and a strong signal. And it's about a 10x improvement in signal to noise when you measure that. And that's super critical if I want a fast and easy way to observe at the nanoscale with my microscope, right? And to be able to capture spectral data at the nanoscale, because good spectral data, regardless of the technique, is always about good signal to noise. And so if you have good signal to noise optics, you can get good spectral data and conduct really good spectral analysis. I'm going to transition now. I talked a little bit about those dark field optics. I'm not going to spend a lot of time on other microscope techniques such as bright field, and reflected dark field optics work very similar, except the performance is not quite as well as they are in the transmitted mode, but I'm going to actually show an example with a larger sample of how well that can work. But also I'm going to transition now and talk about what hyperspectral imaging is, how it works, and what it does. As we mentioned earlier, we can capture hyperspectral data either in the visible near infrared or the shortwave infrared. I also mentioned that we capture this in what's called a line scan mode, which means that we can capture the visible near infrared spectrum from 400 to 1,000 nanometers, or the SWIR spectrum from 900 to 1,700 nanometers. And we can capture that in every pixel of the image. Pixel sizes, depending on the magnification of the microscope, can be close to even 100 nanometers. That's well below, the diffraction limit, right? And so sometimes that's more magnification and smaller pixel size than what's really needed. That potential does exist. you can capture with our technique super high spectral resolution of all of this spectral data, say from 400 nanometers to 1,000 in every pixel of the image. Spectral resolution can be as high as 2 nanometers. If you don't know what spectrum your samples are going to have and how closely one sample element may be to the other, you need the highest spectral resolution you can get so that you can be sure to be able to differentiate those and to be able to identify, for example, a drug load on a nanoparticle, which is possible, and we're going to show an example of that, versus the nanoparticle without a drug load, okay? High spectral resolution can be key to that. The coolest part about hyperspectral imaging, though, as I mentioned earlier, it's the data is presented not only as a spectral curve in every pixel, but also as an RGB image, a red, green, blue image. It's very high spatial context that you get to see the sample in. And it's so difficult for researchers all around the world that are working at the nanoscale, and we see this all the time when we go into laboratories, that there are grad students and postdocs that have hardly ever seen their work after 3, 4, 6 years of working. They occasionally go to the electron microscope and get some limited insight. This is an instrument that's designed to sit on the benchtop in a nanotechnology related lab and give everybody in that lab access to be able to see their sample every day or any day that they need to. And it has incredible utility just from that perspective alone. It allows them to understand how their nanomaterials are interacting with their target matrices. It allows them to understand, if they're synthesizing nanomaterials, how well those nanomaterials are dispersed, how homogeneous those nanomaterials may be in terms of the count or the size. You can see it, and that's a beautiful capability. But also with hyperspectral imaging, you can conduct detailed quantitative analysis, and we're going to show some examples of that here today. The good Darkfield optics, though, are key to that, oftentimes at the nanoscale, right? And so this is a 100 nanometer gold nanoparticle, and there are 2 hyperspectral images. And with an automated stage that we use for the line scan hyperspectral image capture, we have the ability to do registration, to go to the same exact area on the sample, to the same exact pixel, in effect, and have the ability to measure the spectrum based on different conditions. These conditions are identical in terms of the hyperspectral camera, the light source, everything. But here we're using standard Darkfield optics, and here we're using CytoViva's patented enhanced Darkfield optics. And you can see a dramatic improvement in the ability to see these nanomaterials, the same nanoparticle. And you see the optical spectrum measurement is dramatically different, dramatically improved with the CytoViva optics using the same exact hyperspectral imager. So there's a combination that's required of good microscope optics and good hyperspectral imaging to get good data, especially at the nanoscale. With that, we're going to make a transition real quick, and we're going to transition from talking about how the technology works, and I'm going to talk about some very specific applications related to it. And today we're going to focus exclusively on drug delivery related applications, and we're just going to show a few of these and go forward. I'm going to show now something that's not nanoscale. It uses reflected darkfield microscopy with hyperspectral imaging, and we're going to look at an FDA-approved drug. Seretide is the drug that has multiple APIs, which are active pharmaceutical ingredients and an excipient. And we're going to demonstrate how you can identify these 3 elements, the 2 APIs and the excipient, in this FDA-approved drug. Here is a reflected light darkfield hyperspectral image at 10x. And so you can see these are not nanoscale. These are more microscale related sample elements that we're looking here of this Seretide FDA-approved drug. It's an inhaler used for COPD, asthma, and related types of applications to reduce inflammation. And it contains 2 active pharmaceutical ingredients here, fluticasone and salmeterol. And these two APIs are in this drug along with an excipient that is I can't remember the excipient right off the top of my head, but it of course is the predominant thing that we have here. So based on this hyperspectral image that we've captured, this dark field hyperspectral image, we're able to identify the three spectrum that are predominant in this sample, and we see them represented here. This red spectrum represents one of the APIs. The green spectrum represents one of the other APIs, and the yellow spectrum represents the excipient, okay? And so you can see when we conduct spectral mapping based on this spectral library of these three elements, that we're able to map the presence of the two APIs and the excipient, and we cover literally 100% of the sample because that represents 100% of the sample with spectral mapping. And we're able to demonstrate the presence and the location of the APIs and the relative amount. And so for the relative amount, we're able to actually get a class distribution of that. But before we look at that, we're going to look at a zoom image. So now we're up at about 40x, 10x plus 4x digital zoom. And you can see that, you know, there are some differences that just can be observed on a zoom basis. You see this sort of dark area here. Interestingly enough, that's where we're mapping one of the APIs here. And you can see how this mapping matches the geography of this dark area in the sample. There's some other dark areas in the sample here, and we're mapping those as well. You see lots of very bright areas of the sample that we're primarily mapping as yellow as the excipient. But then there are some areas primarily around the edge of this material, and it has, I don't know if you can tell it, but it has almost sort of a green hue to it here. And that's where we're mapping API or active pharmaceutical ingredient 2 here. And I'm not going to try to differentiate which one was which API this morning. It's not the point of this. This is not a scientific experiment. This is more of a marketing explanation of the capabilities of the system. But you can see here where we're able to map these three elements, and you can see some distinguishing differences in the actual sample itself when we zoom it up. But we can also get a class distribution from that mapping that tells us that, as you would expect, the excipient represents the vast majority, more than half of the area that we mapped in this sample. And it's important to note that this is not volumetric data. This is surface area data, okay? So we're getting a surface area, more distribution, class distribution of this, but we're mapping the excipient, we're mapping green for about 28%, and the, API that's mapping green for 28%, and red, which is mapping the other API for about 11%. And we can tell you that this very closely matched the way that this drug was actually formulated. It was a very close match to the amount of API that was expected in this versus excipient. And so this is a non-nanoscale example, but it's very relevant to demonstrate how this works. Now we're going to transition a little bit and go at the nanoscale, and we're going to talk about how that can be done at the nanoscale. And we're going to look at polymer nanoparticles, which are, polymer is a group that is a type of chemistry that we've seen lots of either US or Europe approvals for drug delivery vectors because of the nature of the polymers. And we have two polymer nanoparticle examples here. We have actually a control, polymer particles only, here on the left. On the right, we have the very same polymer nanoparticles, but they have been functionalized with an active pharmaceutical ingredient, a small molecule drug, cisplatin, which is a very common cancer chemotherapy drug. And so when we look at the optical spectrum, example optical spectrum of these two different nanoparticles, we see that the control produces the white spectrum, but we get a very different optical spectrum in certain areas of the cisplatin sample. And it's important to note that cisplatin was functionalized on the outside of the nanoparticle, not on the inside. And it's also important to note that these are two-dimensional images of three-dimensional objects. The polymer nanoparticles are spheres, okay? But this is a two-dimensional image. So if you look at where we're able to map the presence of the cisplatin on the polymer particle based on this unique optical spectrum, we're mapping it more on the outside, okay, of this two-dimensional image. And we're demonstrating that every polymer particle in this field of view is shown to contain the optical spectrum of the cisplatin. And again, we build this optical spectrum of the cisplatin or the optical spectral library by capturing large numbers of spectrum of the positive control, filtering that spectral library against the negative control to move out any false positives, so that we're guaranteed to only be mapping spectrum that's unique to the presence of the cisplatin, which is the only variable change that we see in this controlled scientific experiment, okay? And so we're able to map the small molecule drug on the nanoparticle. And the idea that once you build that spectral library for this, you're able to capture that image in just a couple of minutes, conduct that spectral analysis in probably less than a minute, and know that can be a very powerful information gathering tool for you. Number one, it allows you, if you're trying to develop these polymer particles with a drug or drug delivery, it can allow you to know very quickly how effective are you at getting the API on the nanoparticles. And if I've got 1,000 nanoparticles in a field of view, how many have the API on? 100% or 10%, or what's the number? That's the kind of information that this allows you to get, and it allows you to prove it with an image in a way that's really almost irrefutable. And if you combine this data with other data, such as maybe ICP mass spec or other types of data sets, which may be more quantitative in nature, volumetrically quantitative in nature, but provide no visual proof of that quantitative data. And you combine this instrument, for example, with destructive sample, non-imaging related data. Those are very good ways to demonstrate and prove the efficacy of the work that you're doing. Here we're looking at lipid liposomes that have gadolinium. Gadolinium is an FDA-approved contrast imaging technique. And if I'm able to load gadolinium on a nanoparticle, I'm able to target it much better. And if we look at the optical spectrum, the gadolinium produces over here in the near infrared at about 750 nanometers, it produces this peak that we don't see at all on the control liposome that we're looking at. So this, again, is a way to be able to understand, in the case of this gadolinium-loaded liposome, how many that we have here in the presence of the gadolinium as opposed to the control where we don't have it. Looking at liposomes again, and polymers and liposomes being shown as the first two examples it's not an accident. Lipids, liposomes, the LDL as a lipid, polymer nanoparticles have been the most approved materials that we've seen from a drug delivery perspective. Here we're looking at lipids, liposomes, which have very small sub-10 nanometer gold particles that have been loaded in the bilayer of the liposome, and they appear as green in the liposome when we look at them here, and this is the optical spectrum of them, and so you can actually see it looks like a green circle. That's the bilayer inside the liposome where these nanoparticles are located. And so this optical spectrum is from this green area, and this optical spectrum is from the other, I'll say, yellow areas. And yes, we color matched to what the image sample looks like with the actual spectrum that we showed, just to provide that continuity that we're looking at here. But this allows us to, in the bilayer of the liposome, at the pixel level, know whether or not we have proper loading or not. Here we're spectrally mapping that, right? So again, looking at where they are there, excuse me, and the spectral mapping, we're able to demonstrate the presence of these very small gold nanoparticles in the bilayer of the lipid. And this mapping, excuse me, is a very effective way to demonstrate the loading of the gold in the liposomes. So now we're going to look at looking at some drug-loaded liposomes in vitro in cells, and show how that works. And we're looking here now at liposomes that are on a microscope slide on an isolated basis. And these are liposomes that had been loaded with doxorubicin. Again, we talked about cisplatin earlier, which is a small molecule drug, cancer chemotherapy, and it's a very well-established, approved. Doxorubicin, or DOX, is another approved cancer chemotherapy agent or small molecule drug that's been utilized for years. But both of those drugs, while very effective at cancer tumor sites, are very toxic. So to the extent that we can load those onto or into nanoscale vectors and target those nanoscale vectors directly to the tumor sites, the less toxicity we're going to have around the body. Everybody knows the great possibilities of nanodrug delivery. Here we know that we've got DOX loaded on these liposomes because we get this sharp peak here at about 570 nanometers. Doxorubicin has a natural fluorescence, and that natural fluorescence in the DOX is shown here in this example spectrum of these DOX-loaded liposomes as a result of this super sharp peak that we see here. That's indicative of the DOX-loaded liposomes. This is a very unique optical spectrum, and this is where high spectral resolution can be very helpful, because you see this peak here has a very narrow full width, half maximum. And so we're able to pick that up with great detail in the optical spectrum. And so interestingly enough, when we look at a mammalian cell, this is a live mammalian cell that's loaded with liposomes, optically, you and I with our eyes can't tell the difference between, say, an endosome in the cell and a liposome, right? And one of the great benefits of liposomes is that they really sort of mimic a lot of the natural things that are in the cell environment. And so cell can be very welcoming to them. But if we were just to look at these liposomes against the negative control in the cell, it would be very difficult for you and I to make any sort of, gather the information to say it's there. But boy, we've got some unique optical spectrum of that liposome, which allows us to very effectively spectrally map it with a high degree of confidence in terms of its presence and location in vitro in the cell here. And so this example shows that in a really quantitative way. Going back here now, this is the LDL in the cells that we were looking at earlier. This is the mapping that we do earlier, and again showing some class distribution about the amount of LDL that we actually see here. Here's the negative control again. Now we're going to look at one last sample set, and we're going to look at this in ex vivo tissue. So in ex vivo tissue, where there was an injection of medical grade carbon nanotubes with an API, I think it's cisplatin again, and we're going to look at where the mouse was sacrificed, the tissue was harvested, tumor tissue was harvested, and then looking at nanoparticle exposed and control mice as well, and looking at where we can not only map the presence of the medical grade carbon nanotubes, but also the drug load that are present. And here we're looking at a tissue sample. This is about a 5 micron thick tissue sample, tumor area, where these medical grade carbon nanotubes with, again, looking at cisplatin as the API or the active pharmaceutical ingredient were localized. And we've got bright spots here in the tumor tissue that we can see. And we've got a spectral library here of cisplatin medical grade carbon nanotubes. And this is where we're able to map them, right? But we actually have two mapping. We have mapping of green and mapping of red. We also created a spectral library of the carbon nanotubes by themselves, as well as the carbon nanotubes cisplatin. If you remember earlier, the polymer had a different optical spectrum than the polymer loaded with the cisplatin, and the same thing is the case with the carbon nanotubes. And so where we're mapping red is the presence of the cisplatin, where we're mapping green is the carbon nanotube by itself. And so the idea is that the cisplatin is actually maybe shedding from these carbon nanotubes inside this tissue, but it can be observed there. And we can get a class distribution about how many of the nanotubes alone that we're mapping versus nanotubes with chemotherapy, okay? And here we're looking at spectrum of the nanotubes alone and spectrum of the nanotubes with cisplatin. So, and then here's just a hyperspectral image of tissue that was a negative control tissue. And both of the spectral libraries were filtered against the negative control tissue to ensure that we're only mapping medical grade carbon nanotubes or medical grade carbon nanotubes loaded with cisplatin, again with no false positives, to demonstrate that. So that is taking us back to the beginning. We wanted to make this a very brief webinar. We're about 45 minutes into our webinar, but with that, we wanted to see if we had any questions from any of the participants that are here today. And if we do, Carol, I'll ask you to ask those. And if not, we will conclude the webinar and share the recording of the webinar with everyone. And also know that we would be available if there are questions that you might have specifically related to your research or your samples that you'd like some more information offline. You're welcome to send us. The easiest way is to send an email to us at info@cytoviva.com. We'll receive those emails and very quickly can answer them just with a quick reply, or if more information is requested on your part, we'd also be pleased to engage you with a Zoom or some other method. And I will mention that for laboratories that have a serious interest in acquiring this technology, we can do test imaging of samples. We can have you provide them to our imaging lab for us to do it and review that data together, similar to what we're doing here. But we can also even in some cases, if necessary, bring a demo instrument to a lab if that's an appropriate way to be able to demonstrate the efficacy of the system. With that, Carol, I'll turn it over to you, and maybe we'll go into some of the questions and see if we can answer them.
Absolutely. Thanks, Byron. We did have 3 questions. Oh, we just got a 4th one. Okay, so the first 3 are from Emma Fan. Hi, I missed the first part of the talk, so may I know how fast the technique is, as in the frame rate, spectrum collection speed, and area that can be tested at a time?
Oh no, that's an awesome question, Emma, and I didn't answer, or I didn't really spend a lot of time on that. If you think about this, is a line scan hyperspectral process, and the image that we showed, I'll back out here real quick and just if we look at, I'll go down to demonstrate this. If we look at this image that we captured here, we actually captured this entire image of these sample areas here in under a minute, probably in about 40-50 seconds. If I look at a larger field of view, let's go to this field of view that we have here of the cell structure, maybe a minute and a half or 2 minutes to capture this area. So relatively fast, especially if you think about other spectral techniques such as maybe Raman or FTIR, some of those related techniques. This is lightning fast compared to those. So that gives you some insight. Now, for the optical image, you can have the optical image on your microscope in literally seconds. It may take less than a minute from the time that you plate the sample, put it on the microscope, focus the microscope, and get the image. It's less than a minute typically, once you know how to use it.
Awesome. Thank you. And then next last question. Is it better to build a spectral library by picking up individual spectra or spectra from regions of interest?
It's always, so you're going to map individual pixels of spectrum, right? In each image. And so the spectral library that you build, let's say, for example, that I'm trying to let's go here. If I'm trying to map the gold inside of these liposomes, for example, I would build a spectral library, and I would have two samples. I would have gold-loaded liposomes, and I would have negative control liposomes without the gold. And I can go in and collect large regions of individual pixel spectrum from these liposomes. I can convert that region of pixels to a spectral library of individual pixel spectrum. The software has that capability. I'm not going to try to do a software lesson this morning. And then that may be hundreds of pixels of this sample, and I can filter that against every pixel in the negative control. And that output will be a spectral library of individual pixel spectrum that we can then use to do the spectral mapping. And we can do that with a high degree of confidence. And so that's something that we can get into if we're working more one-on-one with someone, either on site or with your samples.
Okay, two more questions. One from Tima Fernandez. Does the drug have to be naturally fluorescent for analysis when attached to gold nanoparticles?
Absolutely not. No, absolutely not. So the doxorubicin has a natural fluorescence that we showed, so dox is really easy. Cisplatin has, platinum is sort of, one of the agents there. There's nothing fluorescent about it, but it has very unique optical qualities. Even if I apply a decent molar weight of PEG, you mentioned gold nanoparticles, for example, which also can be used as a drug delivery vector. They also can be used for photothermal therapy related. Even if I put a decent molar weight of PEG for biocompatibility, for example, on a gold nanoparticle, what I'm in effect doing is that the active pharmaceutical ingredient or the functional group in the case of the PEG is changing the way that the nanoparticle interacts with light and changing its optical spectrum. So think about it more like, how does the bilayer change, the gold in the bilayer change the liposome spectrum from this to this? Or in the case of the cisplatin, right, how does it change the optical spectrum of the polymer nanoparticle? It changes the way that polymer nanoparticle interacts with light, and it changes its spectrum, but it does it in a highly consistent and highly repeatable way so that we can measure that with accuracy and with confidence.
Thank you. Let's see, Emma said, would you mind to quickly discuss the working principle? How many spectra can be obtained at a pixel point in the around 1 minute detection?
So if you look at, if we go back to this sample as well here, we're talking about hundreds of thousands of pixels of data that's captured in under a minute. So it's really fast. Now, we don't have sample in every one of the pixels, right? But we can analyze that. If we think about the analysis, right? Once I built the spectral library of these three elements, and I conduct spectral mapping, that can be done in literally seconds. It's really fast. So if I go back to, let's go back to this image, this large image of a large number of macrophages. This image capture may have been a few minutes to capture, and I've got, again, hundreds of thousands of pixels in this image, and all of those can be analyzed really quickly. Now, it's going to take a little longer in terms of the seconds to analyze this image versus the smaller image, but you can decide how big you want this image to be based on the image capture.
Okay. One from Feng Li. Can your system see the drug delivery in live, so real time?
Well, so you can do live cell imaging, and in live cell imaging, it is possible, and there have been there's an incredible paper that was published a number of years ago at IIT Madras. The PI is a brilliant chemist by the name of T. Pradeep, and they actually were able to track a nanoparticle in and out of a live cell. So there are live cell chambers. There's a company called CytoViva, which produces printed chips for live cell imaging, and also to mimic an in vivo environment, so that you can look at in vivo, mimic the in vivo environment to look at things like nano drug delivery in real time. But and the way that can easily work, that can be, you can be incubating the cells, take them out of the incubator, put them on the microscope, either capture an optical or hyperspectral image of that, put it back in, and over time, you can look at changes in the progression of these drug delivery vectors and the drug itself. And I apologize, I was going to share an application this morning of spheroids, and I'm not going to try to find that data. But also you can tell from the tissue sample that we demonstrated here, that if you take a spheroid and expose it to nanoparticles or to free drug, you can cross-section the spheroid the same way that we section this tumor tissue here, and conduct the same analysis there as well.
Perfect. Thank you. Jordan asked, we often work with RNA or DNA therapeutics. Can they be spectrally imaged in cells when delivered with polymer conjugates or nanoparticles, or are they too far towards the UV?
So we do have some limited experience. For example, we're all aware of mRNA vectors, these that were utilized for COVID vaccines. And we do have experience looking at lipids with mRNA on it. That's data that we can't share here, but the mRNA will change the optical spectrum of the lipid. It is a difficult lift with this technique, but we have seen some success with it before. That would be one of those applications that we would just have to test to be able to confirm the efficacy. And it's important to note, whether it's this technique or any technique, it doesn't work with all samples. And we want to be quick as a vendor to add that. And so that's the reason why, if we don't already have good experience with samples very closely mimicking what you want to do, we would want to do some test sampling to really ensure efficacy of the instrument for what you're looking for and what you're looking to do with it prior to an acquisition of the system.
Thank you. And those are all the open questions.
Awesome. Well, we appreciate everyone's time today to attend the session. Again, we will provide the recording to everyone if you want to share it with colleagues or review it again yourself. And please feel free to reach out to us if we can be of assistance to you. The easiest way, again, to reach us is at info@cytoviva.com. And we appreciate your time today and appreciate you attending. Thank you.